Please welcome Thomas Durkin for his presentation titled Modeling Neurodegenerative Disorders in 3D Models. Thank you.
Hello, everybody. Welcome to today's talk, and thank you for the invitation to present for modeling neurodegenerative disorders in 3D models. So, to introduce myself, my name is Thomas Durkin. I'm an associate professor at the Neuro at the McGill University, and also director of the Neuro's Early Drug Discovery Unit, of which some of the work we do in the unit I'll be talking about today. So, to introduce a little bit about who we are, actually, we're based in Montreal, Quebec, Canada. And we're in the Neuro. It's an integrated clinical and research hospital. So a lot of the work we do is really focused around who or what patients come to our doors, and then this informs the type of programs that we do, from Parkinson's to multiple sclerosis and so on. At the core of a lot of this is actually a number of core platforms, and one in particular I'll talk about today, our platform, the Neuros EDDU. But before we get into it, I want to talk about a platform upstream of us that helps support a lot of our activities, which is the Neuro CBIG Biorepository. So CBIG for short, it actually means Clinical Biospecimen Imaging Genetic Repository. It's actually, in addition to ourselves, over about 10 years ago, formed as an open science platform to kind of consent patients in an open framework, and also to collect specimens, data from imaging to neuropsych. And this all is then integrated within this kind of LARS framework. And so now at the link here, you can access it, and it's different standards of accessibility are available to everybody that wants to use it. So the reason I bring this up is it actually informs what work we actually do within our group, because the specimens, the lines, they all kind of come because of CBIG, and they actually let us work within our different projects. So within our unit, it's kind of the key to what we do. It's early drug discovery. We actually want to be doing this stuff at kind of the earlier stages. So we're actually working with the patient cells we get from CBIG, from Parkinson's, from ALS. We actually have services and the ability within our group to program these stem cells, to CRISPR edit, to either fix a mutation, to introduce a mutation. And then from this is where we come in. We try to actually build assays using these iPSCs to make the different cell types of interest, kind of for modeling a disease, or even bringing a particular therapy into testing, and eventually then to go back into then clinical trials, or to actually bring a drug into a trial if it works. So just to give you an idea of what we do, actually, we are a high content and automation platform. As you can see here, just to kind of highlight some of the equipment we actually have, from the Image Express to the PerkinElmer Opera Phoenix. It's all automated. It really lets us do all our high content and automated imaging, equipped with a plate mate for compound seeding, and IDOT for specifically kind of drug and dose responses, and then the EL406, which really lets us both handles brain cells and solutions, and actually lets us seed almost anywhere from 20 to 40 plates in a rapid way. So that just gives you an idea of the automation. We also have a lot of the different iPSCs, and our services are kind of outlined clearly within the Navigator CFI funded website that we have. And if actually anyone wants to know more about the cell lines we have that we work with, it is actually available through the cbig-open.lars.ca. But to be honest, like, I'm presenting the work, but this is really a team effort. So people from across different countries in the world that have come to live, work in Montreal with our team, bringing a different array of expertise, from stem cells to CRISPR editing to high content to automation, to really then bring this all together. And this is the people that we work with. And what we do is actually, as a team, we really try to understand kind of three main diseases: Parkinson's, or PD, ALS, amyotrophic sclerosis for short, and then ID, or intellectual disability, and ASDs, or autism spectrum disorders. And we try to match cells of interest to that disease to then both model the disease, or understand, the normal processes, and then eventually then scale it up into a more complex 3D model. And there's really two areas I'm going to focus on for today's talk. On the ALS side, more focus about these 3D motosphere models we're working with and how we can apply them into modeling ALS, and then 3D midbrain organoids for Parkinson's. So to start, I'll go on the modeling ALS in a dish, how we're trying to make motor neurons and eventually get into these neuromuscular, or NMJ models. And to give you a little context on ALS, it's really kind of a disease where you progressively lose motor neurons. And what this means is you're really kind of losing the motor neurons here that are connecting to the skeletal muscle. They're kind of the lower motor neurons, and they connect then up to the upper motor neurons within the kind of cortical region within the brain. But when we started on this, it was really, could we take iPSCs, either control lines or iPSCs we made from patients, and could we actually make the 2D cells, so these spinal motor neurons, and kind of taking protocols within the field, we were able to adapt them to our group, and then routinely now make these motor neurons here, as you can see stained here for NFH, or just the bright field image, really kind of showing the dense axonal network from there. So these can be maintained anywhere from 4 to 8 weeks as needed, and then you can characterize them through a combination of qPCR, imaging, and these just kind of show you some of the images and markers you can get for these different types of neurons. You can actually use them as well to model ALS in the dish. This is just one of the studies from our group. It was actually by a former grad student of mine, Sarah Le Pen, where we actually applied a lot of the different techniques. So we actually took our stem cells, our control line, which we worked with, AIW22, and then we actually introduced two different common ALS mutations, A382T and G348C. And then once we added them, then we made motor neurons and tried to then characterize and phenotype to see what would actually be happening. Were they actually modeling ALS? And indeed, we actually found a specific phenotype that was actually quite characteristic or kind of revealing about what might be happening within ALS. We actually found this hypoexcitability, and it only seemed to happen within probably about 5 to 8 weeks. We did look at some other features of these neurons, from axonal morphology to viability. They did seem to be relatively changed there or unchanged there, and we did see kind of an effect around the synapse. We kind of then said, okay, we now know we can make these iPSC motor neurons. We can actually kind of identify features of ALS with them. So could we actually make maybe a more complex model? And this is really work from another student in my group, Maria Castellanos Montiel, where we actually said, could we make a 3D NMJ model to maybe, study ALS or neuromuscular disorders in a dish itself? And so this kind of came from a project from many years ago where Maria started, where we could actually make the neurons. We could actually get skeletal muscle from primary myoblasts, and we could actually connect them in a dish. What we found was you could actually get bone growth toxin staining where the motor neuron and the actual muscle connected. So you could see these kind of little connections here and here, and you could actually co-stain them with SV2. But one of the problems was it wouldn't let us culture them for very long. So we really wanted to have a system where we could go for much longer, but it was really starting to detach within 7 to 10 days. The motor neurons themselves, the longer you grow them on a flat dish, they often tend to clump attach, and it's not really ideal for doing much longer term kind of culture itself. And you were kind of losing the organization with the myotube. So we kind of asked ourselves, could there be a better system to do this? And so we decided to take this upon ourselves to see, could we maybe make our own NMJ model, almost like in a microfluidic device where you'd have a 3D spheroid, 3D muscle, and then let them connect to each other to form a neuromuscular junction model, taking advantage of the way that motor neurons aggregate together, a system that could allow long-term culture, that we could get this parallel organization within the myotubes, and then we could actually really compartmentalize the two units. And so this is where kind of Maria set off. The first part was really, could we take these 2D motor neurons that we talked about earlier and then culture them into these 3D spheroids? So this is work we published last year in Cells, where we showed we could go from an iPSC to a motor neuron progenitor, and then we could actually kind of mix it together, dissociate to a single cell mix in a new bottom 96 well plates. We could actually make these spheroids anywhere from maybe 4 to 800 microns in size. And then we can actually then grow and mature anywhere from 14 to 28 and even now longer as well. So once you have these spheroids, you can do a lot. You can do a lot of different phenotyping based on what type of techniques and infrastructure you have within your group. For instance, you can just start with the simplest, which is size profiling. You can actually just record bright field images, and through different image J macros, you can observe the diameter, the shape, the size, and this just kind of shows you that as they age, they start to get slightly bigger, kind of consistent with more proliferation and them growing out. You can actually take the spheroids, you can clear them using a technique called cubic, and then you can stain them for some of the different markers relevant to the cell type itself. For instance, here we're looking at HB9, islet1, and NFH. And then we could start to then put them all together and then image them on the opera phoenix. And then we could observe how the different cell types and the numbers and the composition of the spheroids were. Two other things you can do it is one is qPCR. So if you don't want to rely on antibodies. You could do qPCR where you can dissociate and actually look for specific markers, or something we have even ongoing now more recently is you can even do single cell sequencing where you can dissociate and look at the individual cells themselves, or you can actually monitor their activity through a multi-electrode array platform from Axion, where you can grow these spheres here, and then you can actually see how active the spheroid would be within culture. So one phenotype we did actually notice. So we actually, once we had these spheroids growing, we actually did introduce an ALS mutation. It was actually two. So it was in the SOD1 gene. It was actually a G93A, which is commonly used to make a mouse model, and D90A, which is a familial mutation. And we actually grew these spheroids anywhere up to 84 days. So that's 3 months in culture from the progenitor. And we actually found that there was a striking phenotype, and that was that these were actually the mutants SOD1. They were actually hypoactive. They actually were losing activity over time. But the reason why it was actually more advantageous to use, excuse me, a spheroid culture was that it was actually over nearly 56 days that the phenotype arose. If you see here from 56 to 89 days, that's when they actually start to become hypoactive. But when you work with 2D cultures, you only ever really take it to maybe 28 to 35 days. So often you might miss these phenotypes. So it was kind of another reason why when we started working with these 3D cultures, that it would actually let us get to phenotypes that may not become apparent right away. And so this kind of led us to build this project workflow around what do we want to do? Well, we first want to really build the spheroids to scale the muscle, and we'll talk about the muscle in a sec, to then start to combine them into a 3D model, and then to actually build a tool so we could really get the morphological, mechanical, and functional features of this in vitro NMJ model. And once we have that, then to start really starting getting more onto, what is going on with ALS versus controls, and even other neuromuscular diseases as well, depending on the project involved. So we've talked about motor neuron, but what about the muscle? So can you get muscle? And so for this, we actually relied on a company, ZenBio. We were able to actually get their primary myoblasts that are commercial, and then use them to actually then seed myoblasts, and then with an induction kit that they have, go from a kind of myoblast into a myotube, and then kind of grow and maintain these, staying for the marker myosin heavy chain. We worked with a company in Nuvio. So Nuvio actually worked with us to then develop these kind of in vitro kind of biofabricated dishes where you would actually have two wells, but each one had two posts. So that the idea was that we would seed the muscle in, the muscle will grab onto the post, and then you would form this kind of almost 3D muscle elastic band. And then once you had it, then you could start to stain for stuff like desmin as well. So this kind of shows you what you would get at the end. So once you seed the myoblasts in here, then they would go here, and then they start to actually contract. And then you actually start to see the kind of muscle fiber form and really attach around the two anchors here and give you this kind of beautiful muscle skeletal, almost like a band-aid here. And you can stain that then for the different markers like myosin heavy chain and desmin and so on. And this is actually quite stable. We've actually grown many of these over time, and they actually last almost anywhere from 1 to 2, even almost to 3 months, depending on what you then need it for. So once you now have the muscle, you have the spheroid, you have everything you need really to actually make the NMJ. But the question we always asked was, we're making the muscle, but is it active? So this then came back to actually working with Inuvio to actually kind of take the muscle and put them into these devices where we could actually stimulate and record from. And we would then actually give it electrical stimulation. Here was 1 hertz, and you could see with a kind of a calcium knife, fluorophore, you could actually start to see these different activity bursts or kind of twitches here, and then you can quantify with an image J macro in different cells. So it's really kind of becoming apparent that here at the beginning it was a lot more active, and then it starts to really kind of become twitchy, you could say. So how do you then get to an in vitro NMJ? Well, now we have the two ways to do it. We're able to build a workflow where in reality it means that we start to make our spheroid first. Then at day 7, once the spheroid's in production, we start to make the muscle. And then gradually we actually start to put the two into these devices that look like this. So your muscle is here, your spheroid goes there, and this is just a schematic here. And then from there, the neuron or the spheroid actually extends its axon all the way through the channel and then starts to bind and link in to the myosin heavy chain positive muscle. So are these spheroids, are they active within these devices? So to actually test this, we grew it for 4 weeks within the device. And then if you look here, this is the spheroid before it attaches to the channel, and then the channel is where it goes along here. If you actually press play, you can actually start to see is they actually are pretty active. they actually have this kind of almost it's like an asynchronous firing, but it does start to kind of appear like in a networked ability. It does show that a lot of the neurons within these are very active, and this activity then will be kind of hoping to connect to the muscle and start to mediate some control on it. So now we can actually get the pretty much the muscle and the spheroid to kind of connect, forming these NMJs. One of the kind of big things we wanted to really look at is how could we assess, what's the number of NMJs that would form. So we would actually work with a 3D imaging software and mirrors to then do this, where we would actually stain for myosin heavy chain to really stain the muscle, NFH or neurofilament heavy chain, and SV2 for the kind of filaments that are, or the kind of the axons that are descending down from the sphere, and then the bungro toxin to kind of give us an idea of how many are these kind of little NMGs that would form. So this, it seems easy now when we present it. This took almost a year for Maria and others to kind of work out the details where you would really kind of identify these acetylcholine receptors, filter them with the software, then kind of identify the neurofilament and then filter it from there, kind of to then get a readout and reconstruction of what the filaments in these bungletoxin clusters would look like. So it was really more of a sense of what was a real NMJ. And for this, it was really kind of a case of that they would actually be within a 0 to 20 micron range, so very close together. And then the ones that were much further away were not really a neuromuscular junction. It was more just a kind of an axon touching the muscle, but not really forming an NMJ. So now, kind of armed with this type of technique, we can now really get into actually assessing the NMGs themselves. So at the moment, I'm not going to get in for time-wise, we do actually have the ability now to make both iPSC-derived muscle and to actually do different types of morphological and mechanical assessments of the NMGs, both with fluorescent beads, actually, with kind of stimulation, like with the electronic stuff, and also the ability to kind of make this iPSC muscle within a dish itself. And so the goal now is really kind of, which is really the work in progress, is once we now have more of this up and running over the next while, then we can really start to build ALS models, identify phenotypes, and go towards screens for ALS. So the next thing I want to touch on is actually more around midbrain organoids and how we're applying it to studying Parkinson's disease. So one of the big interests within our group is synuclein. So synuclein aggregation, propagation is one of the hallmarks of Parkinson's disease, that you often get these synuclein aggregates that form. And as the disease worsens, as you lose dopaminergic neurons, you have more and more seeds spread throughout the brain. So they can form within the brain, within cells, but you can also make them within a dish or within a tube and actually use them then for different cell assays and also mouse studies. Well, we actually do make our own in-house preformed synuclein fibrils, where they're kind of these fibril-like structures, where they're very long, fibrillar in size. You sonicate them, and then you can use them for different cell assays, or to kind of inform our studies in how these mechanisms are going on with synuclein itself. This has actually led to a recent study, actually not so recent, it's been over about kind of 2 years ago, in Cell Reports, where we found that these synuclein aggregates were rapidly taken up almost within 2 to 5 minutes. It was a work we did with Armin and Dr. Peter McPherson at the and we were really able to show macropinocytosis was driving the intake of these fibrils within to the neurons and cells itself. And there's actually a follow-up paper now that's going to be out shortly within Nature Neuroscience, looking at how once they're there, how they help promote seeding within the 2D cells itself. So that's just kind of to give you an insight into, synuclein is so important when it comes to Parkinson's. So little was actually known about how it even gets in, gets out, and with 2D cell types, we're now really starting to drive some of our better understanding into it. But we kind of had this idea, what about more complex models? We're doing a lot of it in 2D cell types. What about more complex structures, a 3D brain organoid, for instance? And so the reason why we wanted to do this is often 3D tissue or organoids, they're a lot more interconnected. They have a lot of cell types present. They're scalable. You can make a lot of them and maintain them. And there's a real diversity in cell types. When it comes to 2D culture, often you're really working with a more homogeneous culture, or at least what appears like that way, where you only have your neurons and maybe 2, 3 other cell types. But with an organoid, you really have a lot more cell types. And you can see that in a minute or two. And then you can really customize it, the protocols to make different kind of brain organoids representative of different tissues and use this then to test different therapeutics. So we actually probably a few years ago got this up and running, really led by Ngoyan V. Mohammed, who's now moved on, and also Meghna Mathur, where we kind of took our iPSCs and kind of building on some other work already published from Madeline Lancaster, your Noblix group, and many others actually about core cerebral organoid, midbrain organoid, even forebrain organoid protocols going. We're able to take this and adapt it into our group so we can actually make these full brain, midbrain organoids, or MBOs as we like to call for short. And the process is follows where you have your iPSC mixture, single cell, in these kind of U-bottom dishes. They form these, what we call an EB or embryo body. And then you embed it in matrigel, you put it in kind of a shaking flask or a shaking dish, and then you just let it grow. So this just shows it here at day one, quite small in size, maybe 4 to 500 microns, but over 50 days, much longer, they almost reach 2 to 4 millimeters in size. And this is really kind of the original organoids that we actually used to make. Just kind of gives you a sense of their size and also how translucent they really were. So we weren't making them just for fun. We actually had a serious question since we were doing so many of our studies with synuclein, and we wanted to understand we're modeling the processes of how synuclein is getting in and seeding and all of that within a 2D culture system. What about 3D? Is this kind of the same, or are these organoids really not applicable for that? And so V really led this study, and this is the team here. It was published in Brain Communications about 2, 3 years ago, and it showed how in these midbrain organoids, when the gene mutation, triplication mutation was present, where you had excess synuclein, you could really model key features of synucleinopathy within the organoid itself. I'm just going to show you one of the key kind of findings from this is really where we had three different cell types or cell lines, synuclein triplication, an isogenic control or a knockout, kindly provided by Dr. Tilo Kuneda's group. We would find that we'd actually have an excess synuclein being expressed within the triplication line, and this is a lot less within the isogenic. And this is by taking just organoids, lysing them, and doing Western blots. And then the next thing was you can actually see that a lot of this was actually phosphorylated synuclein. So it actually saw over time, about 100 days, you had an increase in phosphorylated synuclein at the immunoblot level. But then when we took these organoids and sectioned them, you could actually start to see these phosphorylated and 129 synuclein start to appear at a much higher level. And the reason why this is important is often the marker for synuclein aggregates forming is on phosphocin 129. So it's an antibody commonly used within the field. And when we were doing this across multiple batches, we started to see it appear more and more again at a consistent manner. But these phosphorylated aggregates forming in both MAP2 positive neurons, but also GFAP positive astrocytes. So this is kind of gives you a snapshot into the paper, but the real take-home message for this is that in a kind of an organoid system, a midbrain system, with excess synuclein, so the first mutation associated with PD, you could see an increase in aggregates form, kind of similar to what you would see within Parkinson's patients. And you would see them in both neurons, in glia cells, and also leading to a kind of decrease in dopaminergic neurons. So gradually we kind of said, okay, now we know we can model PD in addition. Now we have to really see is could we scale this up and make the whole thing a lot more efficient? Could we build actually patient profiles through our patient-derived iPSCs? And to do this, you kind of have to think in multiple levels. Like one is you need to have a lot of different patients. So then once you have patients, you can then compare and contrast. But then you need a way that you can make them at larger numbers and actually be able to do this kind of almost systematically. But you also need the techniques to really characterize them. And that's really going to be the kind of crux of the last bit of my talk is really what are these organoids even made of? What's the jigsaw? What's the kind of the composition of all the cell types? And then what do they look like? Because once you know what they're made of and what they look like, we can start to actually build a kind of almost a unique personalized patient profile on a dish. But before I get into that, we're really going to focus on the techniques we use and how others here on today can really start to learn this and apply it into their workflows. But before we even get into that, to do this, you kind of need a lot of material. Like when we were doing our first study, we were only making maybe 1 or 200 organoids per batch. And for a lot of these studies, you need like anywhere from like 4 to 600 organoids that you could grow almost up to 2, 300 days. And you need a much more efficient process to do it. And it actually led us again to work with the company Nuvio to build this kind of technique published a couple of years ago in Methods, where we made these biofabricated discs. So either with 948, 360, and 137. And that just what that means is It's the number of wells per disk. And these wells are kind of a flat U bottom that really kind of let the iPSCs bind there, kind of form a clump or an EB. And this is really the seed for the organoid. And this is really work from the Magna and Paul that helped put this together. And this just shows you what you have when you're done. You have these kind of 3D midbrain organoids. This is 3 months in these bioreactors that all you do is you keep them growing with constant shaking. And then all you do is maybe once a week change the media on them, and then you just take them out when you need to. So from one batch of maybe 5 to 600, you actually can then have enough to last you for an experiment over 200 to 300 days. But you can even do time points. You can do different kind of techniques, modalities, depending on the questions you want to ask. The other question we wanted to ask is now you have it is could you actually build a kind of pipeline to actually really model and kind of characterize these well? So obviously single cell RNA sequencing is a great way to do it, but it comes with costs. And we wanted to kind of have maybe a more routine way to do it that could let us characterize these a lot faster and kind of get a sense of what was going on from batch to batch. So this is really work from both Dr. Alina Thomas and Julien Siwa within our group to actually build this pipeline. We call it cell type or R, kind of incorporating flow cytometry to really characterize through antibody panels the kind of the complexity of these brain organoids here, so that you could actually build a panel, characterize all the cell types within these organoids, and actually start to annotate, cluster them, and then almost build a single cell profile of them that was actually comparable to what you were getting just based on RNA transcriptomic data itself. So just to show you a kind of a snapshot into how this would look, if you actually take 3 different control lines, and these are 3 different healthy control people, 3450, AJG, AIW, you're actually able to build these cluster maps of all different cell types within these midbrain organoids. And you can see is that most of the cell types found from person to person are the same. You get the different astrocytes, neurons, glial cells. So it really kind of highlights the complexity of these structures, but it also does show you, you do have differences in overall compositions. That's one of the key features we did notice that from person to person, line to line, you do often have the same cell types, but sometimes the ratios and the numbers can vary a little bit. We did a kind of a batch to batch comparison. So we took one of our control lines, AW22, and just looked to see were they relatively consistent from batch to batch. And yes, indeed, they were. It does seem that most of the cell types you would find from batch to batch tend to be roughly about the same, or at least comparable. And you can actually start to see kind of, how different does it look from batch A to batch D, so that when you're kind of trying to do your experiments, you roughly know what you're going to get. And we were able to also build more of a timeline from anywhere from like 38 all the way up to 55 of what the different kind of cultures would look like. And importantly here, these are midbrain. So you actually do have dopaminergic neurons here, and you can start to see it here. So they do actually start to increase in numbers around here, and you do start to see them kind of decrease a little bit the longer they're there. But that's kind of, they do have a lot of the other features there, and it's kind of similar to a midbrain region where you would maybe only have 15 to 20% often within the midbrain region within the substantia nigra. So one of the other things that we can detect the midbrain and the dopaminergic neurons within these organoids, what we kind of wanted to know is were they functional. And to do this, we kind of had to rely on almost like a neuromelanin imaging process that we worked with Juan Aguera in Munich to really develop along with Ludwig and Maria. And the idea was, could we take these organoids in 3D and actually through a optoacoustic mesoscopy, kind of image the neurofilaments or neuromelanin within the organoids themselves? And you could actually indeed see it here. There's a feature of these midbrain organoids is one is if you give them dopamine or L-dopa, neuromelanin forms, or you just age them for anywhere from 5 to 6 months, the neuromelanin also forms again. So it just shows that the neurons or the dopaminergic neurons in particular are functional within these 3D structures. And then you can kind of image them and stain them here across with this different technique. So kind of the last part is now we can image them. We know we have neurons, different dopaminergic neurons, astrocytes, glia. How do they look like? So this is a bit more technique focused, but we wanted to kind of highlight some recent work really kind of pioneered by both Maria and Martin within their group, and in parallel with the Serbo group in Laval, where we're kind of taking these AAVs or adenoviruses and actually kind of co-infecting our organoids at different months. Here, we'll give you an example of a workflow for 6-month-old organoids to show how you can both infect, stain, and actually do quantification of the different cell types within the kind of 3D organoid or structure. Just to show us what you can do is so you can do it live, You can do it fixed. You can do it after under 5 days of infection. You can do it after 1 month. And you can really kind of start to build a 3D rendering of it with the mirrors of the whole kind of 3D structure that you would optically image through on the organoids. And once you have that, you can actually start to focus in on regions of interest with astrocytes, neurons, and then restart to build almost like an arborization map and kind of identify, how many neurons are there, how are they connected, and get a sense of the connectivity within the structure itself. So just to show you kind of what this looks like, you have both your real time and your fixed organoids. The real time is on the left, the fixed is on the right. It kind of just shows you the different complexities, the different cell types. So GFAP and synapsin are the two markers we use with AAVs. GFAP marks astrocytes, so the green cells are actually astrocytes, and synapsin is neurons. So you can actually start to see the complexity of these neurons. We already showed it to you on the single cell way, and this is more showing it to you on the imaging way. But it shows that you can do this both in real time and then here on right. This is actually cubic cleared, and you can start to see a little bit for TH and Hoost as well. So now we know we can do it. How does this whole process work if you want to do single cell phenotyping of cells? Well, once you build your 3D construction, you kind of see it looking like this. And this is really more true, almost like half of it. You start to build your masks. So you can actually start to demarcate different channels, either for GFAP here would be astrocytes, for neurons will be in gray. You can start to show and highlight which cells go where. You can start to see how they connect to each other. And then you can actually start to zoom in. So you can really start to see which cell type is which. You can say, okay, that's a neuron. How far does it extend? What type of volume does it have? And so on. So then you can really start to build almost like a single cell map based on the actual cell types present, and also through these imaging tools. So for instance, you can start to build these neuronal area maps. You can really start to kind of demarcate all the neurons here. So this is synapsin positive neurons. You can kind of detect the volume of neurons, the area they overtake, and you can get different examples of how much percentage do they take compared to GFAP positive cells. You can actually now zoom in on specific cells. So we actually zoomed in here on this particular cell type. You can see how much surface area all the connections will form and get an idea about the volume, the length, terminals, and so on. And you can even start to look for branching. So if you even look at a different site, you can kind of look at individual cells and how they compare against each other. So you can almost start to do this and profile that at a single cell imaging level. So this is really where we're at so far. Kind of wanted to highlight two areas we're trying to go to with ALS and Parkinson's, where you can actually, through cohorts of patient lines, you can actually start to build the tools, the models, to really start to understand what signatures may or may not be emerging. And the two key features or techniques to really keep in mind when doing this is, what do they look like? What's the composition? And what are the imaging tools you can apply to then actually see what do they look like? And ultimately, the goal is really to come back to this, what we talked about at the start, where how do we start to model disease in a dish? Like to go from these simple 2D assays, which are coming from the cohort of iPSCs you made, and then starting to get into these more complex models from 3D organoids to 3D NMJs. Once you now have them going on, and you have the ability to both bring in kind of a combination of imaging tools with transcriptomic tools with morpho kind of metric assessments, you can really start to assess compounds, and ultimately the goal will be to kind of bring some of these candidates into the clinic in the next kind of decade or so. So I'm going to end here. If anyone wants to know more about the work we do within our group, this is just kind of a flavor or a snapshot for now. I'm happy to have your attention today, so I'm happy to take any questions or comments, and hopefully you enjoyed it too. Thank you.
All right, hello everyone. First of all, thank you, Dr. Durkin, for your interesting presentation, and thank you to the sponsor of the webinar, Sartorius. We're about to start our Q&A session, and we already have a few questions in the chat. If we don't have time to get to all of them, Dr. Durkin will contact you by email with the answers. So let me get one here. So while culturing more than one cell in the same plate, how will the media for each cell line be given? While culturing more than one cell in the same plate, how will the media for each cell line be given? Oh, sorry to interrupt. Your mic is muted.
Sorry, my mistake. So I guess if you're doing co-cultures, often you just make the media. You often have a hybrid media that you would make, and then you just add it as you remove the media, add it back in again. When it comes to organoids, often they have their own growth media that they're maintained with all the factors, ingredients. So you just change it once a week. You just take it off, add it back on again, and let them grow. It's not too much magic, I'm afraid, to yeah.
Excellent, thank you. So this one is, I think, a two-parter. So I'll go with the first one. So firstly, thank you for the interesting talk. I was wondering if you observed necrosis, given that the embryos were at times cultures for several hundred days. And that's the first part.
So when we first did these protocols many years ago, we did. So we used to do these in orbital shakers. They would be like 5 to 6 organoids per well. They would be shaking around. They were a lot bigger in size. So they'd be anywhere from like 2.5 to 4 millimeters. You'd often see a lot of dead cells in the core that vary in size. We don't see as much issues with viability now. We can often get organoids anywhere from like 90 to 95% viable, depending from line to line, and often if it's a healthy versus a disease. The reason we find is it's actually because they're a lot smaller. So we've often then made them smaller, but they still have almost the same cell number. So then the media within the bioreactor, because it's shaking, can actually pass a lot more freely, kind of helps in maintaining their viability. And this lets us kind of really grow them for hundreds of days. So it's been kind of a couple changes almost in a way. By scaling up to a bioreactor, it made them healthier, smaller, and then they weren't banging themselves against the edge of the well when they're not in a six well anymore.
Excellent, thank you. And this is the follow-up to that, which is, and similarly, did you not encounter issues when culturing the 2D cultures for up to 42 days?
You do. One of the big common issues we have is often the neurons like to either dissociate or clump. So that's why a lot of the assays we would do often tend to be between 4 to 6 weeks. When you go longer, you have to find it's this higher density to keep them there, but sometimes it makes it difficult to image. And then when it comes to motor neurons, it's often that's why we kind of switched to spheroids. We kept noticing that they would clump after 6, 7 weeks, so we could get them to an MEA, but then by clumping, it would made it difficult to image. So we're like, well, if they're clumping, why not just make a 3D culture? And then lo and behold, now you can maintain them for longer and clumping doesn't really matter because that's actually
Excellent.
what you want. You want an aggregate.
Yep, yep. Perfect. Thank you. Hello, Professor Durkin. Thank you for the talk. I was interested to know whether the lab also works with four-brain organoids, and if so, do they follow the growth patterns and time period?
We do. We do a lot of work on developmental biology. We just didn't have a chance to talk about it today. So we did have a recent paper with Fragile X. That was more on 2D cortical neuron side, but we have made forebrain organoids. We tend to follow Sergio Pasca's protocol. Seems to work quite well. So it's rather than shaking, it's more static cultures. We're yeah, we've grown up some batches, maybe not as much for extensive studies, but kind of followed his workflows, and it seems to work pretty well in our hands.
Great, great. Thank you. Can steroids be implanted in the brain?
And um, steroids be implanted in the brain. I guess you could certainly inject a steroid like hydrocortisone and stuff. I guess it really depends on your scientific question and what you would want to do. Hormones, I guess, are around the body. They're in the bloodstream. They do pass through it. So yeah, it would all depend on your scientific question and whether you want to do that or not.
Yep, sure. Yep. Have you worked on Alzheimer's with these techniques?
No, so we haven't. The reason we really focus on Parkinson's and ALS is really those are the main cohorts we see within the institute itself. We do have 1 or 2 projects that are going to start soon on Alzheimer's. We do have one project where we're looking at seeding and aggregation in neurons, and then another one where we're going to start looking at some of the Alzheimer's lines. But often because we don't have the patient cohorts readily available, we have to go outside to get them. But we do have some stuff to start, but nothing really ongoing at the moment. That'll probably be coming over the next few years in terms of that side of things.
Yeah, that's great. So, Why 3D and not 2D cultures? What's the key advantages in your mind?
I think the advantage is timing. You can actually grow 3D a lot longer. So, two features we noticed is with 2D, let's say neuronal cultures, you can grow them a lot longer, and then you can actually start to see activity defects. Sometimes when you grow them for a short period of time, you might miss features that are coming with longer maturation. And then the other aspect is kind of highlighted with the organoids itself or midbrain. You actually get neuromelanin that forms. So you get a maturation that you often don't get with the 2D cultures. So they start to kind of become tired, don't like being attached, and then they start to die off. Whereas with a midbrain organoid, they can grow and grow and grow. So you can really capture more of the features and synchronicity of what you may see with a brain as it develops. So it's really the longevity that you get from it.
Yep, yep. So, speaking of 3D motor neurons, any tips or tricks for getting them to attach to the Axion MEA multi-well plates?
I think sometimes and often you have to play with the cell density. We often find that neurons prefer to grow on the axion plates almost better than imaging plates, but it does take a little bit of trial and error in terms of how well you seed them, kind of some of the conditions, and you have to always be careful then of the medias and kind of the recording media you use once you're doing the measurement itself. So I think it's really a case of if you're trying a new cell type for the first time, just maybe play around with some of the densities, maybe have one or two separate cell lines so you can compare and contrast, and then eventually you'll kind of optimize it. And hopefully then, yeah, you'll want to see, in a way, firing. Like if you don't see firing, then you might have a problem. And make sure you have access to some of the well-known EFIS chemicals that people do use in the field.
Yep, yep, perfect. Thank you. Do the iPSCs from ALS patients grow similar to or other neurodegenerative diseases and or normal donors and have similar phenotypes?
To be honest, if you grew a lot of different motor neurons from patient lines, you wouldn't actually notice much different just by like most of them grow motor neurons. You can see them and look at them after 3, 4 weeks and they almost look comparable. So that's kind of where some of the issues arises. You really have to start digging deeply and make the model maybe a little bit more complex or start to bring in some maybe maturation, more advanced modeling, transcriptomics. So for example, like the mutant sod line versus sporadic line, they all form motor neurons. There's no real viability defect for the first few weeks. So it's really more you have to add a stimuli or a increase maturation or kind of make it something a little bit more advanced to get something.
Perfect, perfect. Thank you. Okay, this is a fairly long one. You showed some nice CA firing, which I believe was from 3D models. However, it seemed to lack some synchronization that is usually seen in 2D cultures. Is this a common observation you have in 3D, or does this improve with the maturation slash age?
I think it will improve. This is really just a proof of concept that the spheroids we were putting in the dish could they were active, so they were still quite young, maybe 2, 3 weeks. I think they will improve and become a lot more synchronous if we got it to like 7 to 8, 9 weeks. So I think that's the only caveat with that is they were still a little bit maybe on the immature side. I think what we were really going for is are they active or not? So they did seem to be active and all of that. So I think synchronicity will come a bit later on. The other issue with these were they are only one cell type, they were only motor neurons. So I think, if we start making these more complex, adding in glial cells and that such type of cells, making them more complex will actually help with the synchronicity as well later on.
Yep, perfect. Thank you. The developed organoid 3D models subjected to screening for the drugs or for small molecules. Is it feasible?
We believe so. That's kind of why we've been building it. So I think we spent a few years trying to make sure we had the models so robust we could characterize it. So that's actually where a lot of our energies are going into now. Like we have a few candidates, both small molecules and RNA therapeutics that have come from the 2D side, and we're now going to be advancing into these 3D models to see if there's features that go from a disease back to a control or even a rescue. And yeah, watch the space. That'll be where we're going in the next few years on it. But yeah, that's pretty much what we want to do with it.
Yeah, that sounds really interesting. Thank you. Um, what have been the biggest challenges in getting these models set up?
Time. A lot of the stuff is time and sterility. Often you grow these for hundreds of days, and one thing goes wrong on the one day and the last day, and you get contamination, and your whole culture is gone. So you have to really learn very careful planning how you build them almost in batches, stagger them, make sure you've got a very clean and sterile environment, well-trained personnel, which we're fortunate to have, and really then all the various stuff then to go in. So I think that's the key. Is it's often patience. People think that they're working with a cell or helicell. That's not the case. These are complex. You need to understand the cells. You need to understand the methods, and then really have the advanced characterization to go with it then as well. But just be patient and give it a few. It's not going to be in weeks. It's probably likely months to years before you really start to see the concrete efforts of your work emerge.
Yep, absolutely. Yep. So which method or technique is best to make brain cells from pluripotent and multipotent stem cells in terms of treatment with certain small molecules or transfection?
I think that's really dependent on the disease you want to study. We find you can make any cell type you really want depending on the molecules, but it's all the question you have to ask is what is the disease and what cell type do I think is most kind of important to the disease, and then go from there. So often we have like 2 or 3 major control lines we use a lot, but you can direct them into dopaminergic, cortical, motor neurons, astrocytes, oligodendrocytes. The list goes on and on. And there's others in the field and other labs that have similar cell types. So it's not really a question of is small molecules, is there one protocol better than the other? I think it's, or a lot of protocols that work, but it's really what is the question you want to ask? And is it a developmental question, a disease question, and what's the most pertinent cell type you should work? And don't always think it's a neuron either. Sometimes it could be a glial cell too.
Yeah, perfect. Thank you. Can we co-culture neuron and myoblast stem cells in hydrogel scaffold?
It's potential to do so, yeah. It's not something we've been trying to do. There might be others that have done it. I know there's I think it's from Pasca's group. I know they have been doing stuff where they co-culture a lot of the organoids more as assembloid models. So that's been a kind of common thing to do. We steered away a bit from that because we wanted to have more of a separation. But we know people have put like a cortical, spinal, muscle all bound together, and I guess they can put them all together and grow them for several months as well. If you want to try hydrogels, there's no reason. We have done work on, I think it's cortical organoids back in the past with alginates just to model stiffness, and it's worked quite well. But it really depends on if you have the time and want to try it. So if you've got the model up and running and you want to experiment, I say go for it.
Yeah, perfect. Thank you. So somebody says, thank you so much for the insightful lecture. I want to ask how CRISPR-Cas9 plays a crucial role here.
Yeah, we didn't really talk about it. Like we actually do have a few people that actually do this routinely. CRISPR's been game changing for us because we made a lot of knockout lines, so it actually helps us knock out many genes and study the effects of given genes in a disease. We actually, a lot of our patients are monogenic form, so you often have a mutation, so you need the control to go with it. So we could actually use CRISPR to correct the mutation. And there's actually been a few cases, in particular one with a SOX10. It's a reporter for oligodendrocyte generations. We were able to add a tag onto it, so we can actually really identify and enrich for a progenitor population. And actually kind of allowed us to improve the protocol we had. Like we were getting very low yields in oligodendrocytes. Once we had the reporter system, we could really start to enrich and get the cell populations we have. So I think if you're thinking about adding CRISPR to your workflow, say yes, do so. Just make sure you have there's a few pieces of equipment you need, and once it's up and running, you'll be able to kind of make sure you have the appropriate controls. You can make reporters and really do a lot of things and ask a lot of important questions.
Yeah, that's great. That's great. Thank you so much. Some really clear answers there. That's actually all the questions we have. So we'll wrap up the webinar there. Thank you all for attending. Thanks again, Dr. Durkin, for joining us, and thanks everybody for watching. Have a great evening, morning, or afternoon, wherever you are.
Have a great day and week and everything for everybody. Nice to see you. Take care.